Welcome to the transcription in prokaryotes and eukaryotes. To begin to understand how DNA makes a specific protein, you need to understand how transcription works and the process of what you have to go through to really, make your specific protein. So in more or less words, transcription is the process involving RNA molecules and they synthesize on a DNA template strand. The template strand is chosen when the RNA polymerase comes through, but we'll talk about that later. Transcription is an essential process because it's the first step in how information travels within a cell. So here we have a bacteria cell, or also known as a prokaryote. And within the cell, there's a whole bunch of different regions, like the cell wall, the nucleoid region, the plasma membrane, the cytoplasm, the ribosome, but we'll mostly be taking a look at the nucleoid region. And here we have a double helix. And this is going to be the first part of the template binding. So right here you see a promoter site, and the promoter site is needed because it lets the sigma subunit on the RNA polymerase, which you'll see here in just a second, attach and latch onto the DNA template strand. And it's actually known that the RNA polymerase will travel up and down on the helix to find this promoter site so it can attach and start doing its job. You also see two types of consensus sequences. The first is the TATA-AT sequence. It's located 10 nucleotides up from the started transcription site. And the other one is the TTGACA sequence, and it is higher. It's even higher than the first one in that it is 35 nucleotides up from the started transcription site. In eukaryotes, there's a region much like this, and it's called the TATA box because it's so rich in adenine and thymine. Get it? TATA, thymine, adenine. So anyway, once the sigma subunit, which we're seeing right here, on the RNA polymerase attaches into the promoter site, this is going to start initiation. When it attaches, it's going to be a closed complex, and when it opens up the double helix, it's going to be an open complex. The initiation process is a vital part in this whole role because it attaches the first 5' ribonucleotide triphosphate, and it'll complement like the start side of the DNA template strand. So this is going to be, this right here is the template strand, and it's going to come in and connect there. And then after that, the rest of the floating nucleotides that are around and floating in the cytoplasm are going to come, and you'll see the strand. So here we see all the different molecules consisting of A's, U's, G's, and C's. They're going to be complementary to the DNA template strand, which is already there, and there's a little preview for you. Let me go back and the A's, the T's, G's, and the C's. But then it's going to start changing right here with the U's and the A's because in bacteria it uses U's instead of T's. Keep going. And then as this is going, it's making an elongation of all these different molecules. And this elongation process is going to keep going until they reach a stop codon or a termination point. And sometimes they have a little trouble coming to like a stop, so that's when a termination factor Rho comes in. And Rho is a protein that works with the ever-growing RNA chain, as you can see right here. The RNA polymerase, along with the sigma subunit, will eventually detach, and the Rho will form a stem loop, which you see going on right here, and help detach the RNA strand. and everything falls away, and there you have your RNA strand separated from the double helix. And here again is the row. You're going to see this here in just a second. The row factor is going to attach to the RNA strand, move up, and then it'll make the stem loop. The ribosome will fall away, and the whole thing will detach. So again, what you're seeing here is termination. It's reach and stop codon. Sometimes the row factor has to come in and help discontinue this whole thing, and then it comes to a complete stop. And now we'll move on to like the differences between a eukaryotic and a prokaryotic transcription. One of the main differences is that in prokaryotes, they move to the cytoplasm to carry out transcription, whereas eukaryotes can stay in the nucleus. A second difference is that eukaryotes have enhancers, which are like promoters because they control transcription processes, and we'll talk about that in a little bit. And the third difference is in our cells, or eukaryotic cells, we go through processing where a 5' cap and a 3' tail are added to the end of our mRNAs, or messenger RNAs. We also have larger molecules in our nucleuses, like pre-mRNAs, and a whole bunch of, and when you have a whole bunch of pre-RNAs added together, it's called a heterogeneous nuclear RNA. So transcription in a eukaryote, which is like me, you, plants, and animals, is a lot like a prokaryotic transcription in their cell. Eukaryotes have a promoter region, like you see here, but sometimes it's aided by the help of 3 cis-acting elements. For example, there's like a Tata box, like I said before in the last video, which is 30 nucleotides up from the start transcription site. And it gets its name because there's an abundance of adenine and thymines, AT. There's also another region called the CAAT. It's even farther up than the Tata box because it is 80 nucleotides, my bad. This helps with the efficiency of the promoter. So that would also be around or up farther in this region. And also in eukaryotic transcription, we have enhancers, which is most likely to occur the farthest upstream from the transcription site. So whereas we would have like the Tata box here and the CAAT here, the enhancers would be even farther up in this region, or they're like embedded other places along the site. We also have transcription factors that are needed because RNA polymerase II can't latch on directly to the promoter site without them. So that would be right here on this picture. So just like in the prokaryotic transcription, an RNA polymerase would come in with the sigma sum unit. The sigma subunit would split the double helix as shown in this picture, with the RNA polymerase still synthesizing at the end. The free molecules, the A's, the T's, the G's, and the C's would all come in, and they would make a complementary strand of what the DNA has already had, and it would keep going until they reach the termination codon right here, and they would be off into their own thing. And so like I was saying before, where prokaryotes can move to the cytoplasm, the eukaryotes can stay in the nucleus and make this just fine. And this is the closing of the transcription room. We really emphasized the prokaryotic transcription and how that all worked out. And then at the end, we learned a little bit about eukaryotic cells. Now you're going to be moving on to the translation room. See you then.